start the class, right? Okay, last class what আমি কি কৈছিলো, we were talking about the male reproductive part, that is the anther. And what we have studied is, we have talked about what is inside the anther, no? Because we need to study something called as microsporogenesis and megasporogenesis, since we are talking about sexual reproduction, and because of that we need to study the anther, and obviously we need to study the carpel also, right? Starting from the anther, we have always for our study what we choose is a immature anther ne mature anther, immature anther. Immature anther কিহৰ কাৰণে choose কৰো, so that we can see the process of development of the pollen grains properly, right? So whenever we choose a immature anther, what we have done is we have cut a TS of the anther, and when we have cut the TS of our anther, we have seen a lobed structure. We have already discussed the terms called as bilobed, or right, bilobe structure. We have talked about bisporangiate, tetrasporangiate, etc. Which one is most common? Obviously tetrasporangiate in case of angiosperms. So TS eta jeta katiba, you will see a lobe structure, and from the diagram that we have drawn, what we know, the outermost layer is called as the epidermis. Below the epidermis we have the hypodermal layer, which is also called as archisporial layer. Out of the cells that are present in the archisporial layer, one cell in each lobe will become big, or tahante dangor hoi jabo, and that cell would be called as what? Archisporial cell. Archisporial cell will divide into two cells. What is the type of division they will undergo? Anticlinal ne periclinal? They will undergo periclinal division, and ultimately they will form two cells. And what were the two cells? Inner primary parietal cells and inner primary sporogenous cell. Primary parietal cell will form the cell wall, it will form the pollen sac or wall, which is, we know that it's almost about 2 to 6 layered thick, and the sporogenous cell will ultimately form in the process of development ki form koribo, pollen grains kini form koribo, right? Next is, we have started with the process called as microsporogenesis. What is microsporogenesis? From that sporogenous cells, the process of forming the microspores is called as microsporogenesis, no? MMC is a word that we use. We can also use a word called as PMC. MMC hole microspore mother cell, PMC hole pollen mother cell. Eketai bostu. What is the ploidy? Obviously twice-cell, right? And that cell will undergo meiosis and will form a collection of 4 cells, and these 4 cells would be called as what? Microspores. Microspores is equivalent to ki? Pollen grains, right? Egida terms to clear, no? Microsporophyll, microsporangium, microspores, right? Microsporophyll or ploidy ki? Twice-cell. Microsporangium or ploidy ki? Twice-cell. And if you are talking about microspores, what is the ploidy? It is N, right? Diploid, haploid, we understood. Last class on what we have talked about is the formation of microspores, and we have seen that there is a collection of 4 microspores that are formed, and these microspores remain attached with each other, and they are arranged in a particular pattern, right? Talking about plant to plant, no? Different plants or ami a arrangement to beleg dekha pao, different plants auto dekha pao, and different groups auto dekha pao. If you talk about a monocot, you will see a different type of arrangement of the pollen grains. If you talk about a dicot, you will see a different arrangement, right? So these 4 pollen grains are not arranged in the same way in all the plants, right? So today we are going to continue from that, right? So likhi by arrangement of pollen grains, by arrangement of microspores ji likha. Let me write it down first, okay? No, no, এটা চার্টখন কমপ্লিট তাৰপিছত তলত লিখিবা দে ঠিক আছে নালাগে নালাগে সাইমালটেনিয়াস হৈ যোৱাৰ পিছত তলত লিখি ল'বা ইয়াতে নোটছত নে ডায়াগ্ৰামত ডেকুচেট DECCUSATE over হৈ গ'ল okay so what we have said is we know that from the pollen mother cell or microspore mother cell the microspores would be produced by a process called as meiosis. Obviously reduction division hoboi lagibo because we are talking about the reproductive part of a cell. Now talking about meiosis, no, meiosis or mitosis, whatever we are talking about, it is always it is always of two steps. We have karyokinesis followed by cytokinesis, right? So whenever we talk about cytokinesis, so this particular arrangement of tetrad always depends on what? Always depends on the process of cytokinesis, and out of the entire note that we have written, only this and this arrangement is something that is asked from the examination point of view, right? All you have to know is the word isobilateral and tetrahedral. Questions kya hai? The isobilateral arrangement is a characteristic of, tetrahedral arrangement is a characteristic of, which one is most advanced, etc., etc., right? Either the arrangement would be isobilateral or it will be tetrahedral, depends on what? The process of cytokinesis. So yato cytokinesis hoy, tato cytokinesis hoy. What is the basic difference that we are writing it down? What is the difference? Isobilateral arrangement hobo lagile ki hobo lagibo, tetrahedral arrangement hobo lagile ki hobo lagibo. We have written a line called as cytokinesis occurs after each meiotic division. What does this line tells us? Meiosis is kiman parts of divide korise meiosis to? Two parts of ki asile? Meiosis 1 and meiosis 2, right? And whenever we say that the cell is undergoing meiosis, we understand at the first stage ki hoy, the nucleus gets divided into two. What next? Eta cell jedi meiosis hoise, then what is the first step? One nucleus get divides into two, right? Prophase, prophase 1, metaphase 1, telophase 1, etc., etc. After that first meiosis, what is the event that is followed? Meiosis or eta cell keita offspring produce kore? Saita. So saita produce koribo lagile saita nucleus produce koribo lagibo na lage? Lagibo. So saita nucleus keneke produce kore? Is it like this that one nucleus will get divided into two? Two nuclei will again get divided into two, that will make a collection of 4 nuclei, and then the process of cytokinesis will start. That is one way. The other way around is one nucleus divides into two, cytokinesis starts, they will form two individual cells, and those two individual cells will again undergo meiosis 2. Which one is correct? The second one. Is the point clear? Right? Whenever we are talking about meiosis, we understand that one cell will obviously produce 4 cells, but production of 4 cells will involve 2 types of cytokinesis or one cytokinesis. It is 2 cytokinesis that is important, or it occurs, right? If meiosis 1 is completed, after meiosis 1 what occurs is cytokinesis 1, then meiosis 2, and after meiosis 2 cytokinesis 2. If this is the process, then that type of arrangement would be called as isobilateral, and this is supposed to be the most advanced. So this is a point, this is a point, and this is a point. Whereas in this case, what we have written, cytokinesis occurs after complete meiosis. Complete meiosis mane one nucleus dividing into 2 nucleus, 2 nuclei dividing into another 2 nuclei, collection of 4 nuclei, and after that only cytokinesis has taken place. Either this is the process or this is the process. Normally which process we talk about? We talk about this process, right? So eneke jedi hoy isobilateral arrangement hobo, cytokinesis jedi matro ebar hoy tetiahole arrangement to ki hobo? Tetrahedral. So which one is seen in dicots, which one is seen in monocots? That's important, right? As far as these are concerned, no, these are certain extra things. We don't have to think that it's only isobilateral and tetrahedral is only seen in case of plants. We see different types of arrangement in specific plants. There might be some other examples also, right? Next, as far as the meaning of these words are concerned, diagram we have done. So please look into the diagram carefully, especially isobilateral and tetrahedral. Isobilateral, tetrahedral, what is the basic difference as far as the diagram is concerned? Isobilateral, tetrahedral, what is the basic difference? Okay, the first point is, if you are talking about microspores, 1 mmC will produce how many microspores? 4 microspores, hetu to sure. So each and every diagram, what you can see, a collection of 4 microspores. What about tetrahedral? 3 microspores. Tetrahedral mane 3 ta niki? 4 ta aru eta kot thakibo? Backside, right? So if you can see all the pollen grains at one face, then the arrangement is called as isobilateral. If you can see just 3 in front and 1 at the back, then it's called as tetrahedral. Obviously this would be a 3D diagram, so yat to are possible nohoy aki bole. So from this entire discussion, important key, successive isobilateral most advanced monocots, similarly simultaneous tetrahedral dicots. Clear? So this was all about microsporogenesis. After microsporogenesis, now we know that the microspores are ready, right? They are formed, but germination etiao hua nai. We'll be talking about how does the male gametophyte develops. But before talking about that, we need to write certain points. Points boli likhasu. Some important points, okay? Online students clear? Okay. Okay. এইখিনিত প্ৰথমে লিখি যাওঁ। Is it over? Okay. First come to this point. Wholeness over a whole. Fast. Akilu. Okay. So, certain points that we have to write. First, let's come to this word. As we have already discussed, we know the MMC or the PMC will undergo meiosis and will form a collection of 4 microspores, and all the microspores would have a particular type of arrangement and they will be arranged together, or that would be a compact structure. Before the process of pollination, each and every microspore will have to separate from each other, and only after that pollination hobo. Right, the microspores of the tetrad separates from one another. Up till here we understood. Next is germination in situ. Right, so question ahe, microspore germination is in situ ne ex situ. Now you have to understand what is in situ and ex situ. What is in situ? Inside something. Inside what? We are talking about the germination of the microspores, right? Microspores will germinate in situ. That means what? The microspores will germinate inside something. Inside what? Inside the microsporangium or inside the pollen sac, right? So pollen sac to bhitorte, we remember we have talked about that anther. Inside the anther we had those 2 cells, no, the wall, the microsporangium, etc. So tar bhitorte jodi microspores germinate kore, and what we understand by the word germination of a microspore? Microspore ki hoy? Pollen grain. Pollen grain to kihor bhitor germinate korise, pollen sac tur bhitorte or microsporangium bhitor germinate korise. So we are using the word in situ. The next point is what you understand by the term germination of microspores. Germination mane ki? In regards with microspore, when we talk about germination, normally we think about the germination of a seed into radicle and plumule, but here we are using a word germination for the pollen grains. So what exactly is the meaning of that sentence? Germination boli kole seed germinate kore, no? Seed germinate kore ki produce kore? Radicle and plumule. This is something that we know. Kintu yate ki kose? Pollen grain or germination means what? Pollen grain is just one cell dividing itself into 2 cells is the process of germination. Remember the words called as vegetative cell and generative cell, tube cell and generative cell, no? We have one cell as the microspore. This microspore will divide itself and form 2 unequal cells, and this process is what is referred to as germination. Clear? Okay. Next, in Typha and Drosera, the microspores does not separate. Normally what we know, normal cases ki hoy, microspores separate kore before pollination, but in these 2 cases what they form is something called as a compound pollen grain. What is a compound pollen grain? Where the microspores do not separate and stay together with each other, that is what is called as a compound pollen grain. Question kiya hai? Compound pollen grain is a characteristic of, but Typha or Drosera ki dekha pai. So especially these 2 examples and the word compound pollen grain, right? Next, in these 2 families, especially Asclepiadaceae and Orchidaceae, we see a structure that is called as Paulinium. Akko question ahe, Paulinium is a characteristic of which family? Paulinium is a characteristic of, specially question ahe Orchidaceae uporot, right? Orchidaceae nidi palai. In the option, orchid could also be written, not necessarily the name of the family would always have to be written down, right? So instead of Asclepiadaceae, in the option, Calotropis could also be written, right? So Calotropis are ami ki dekha pao? Paulinium. Orchids or ki dekha pao? Ami Paulinium. Now what is a Paulinium? So all the microspores of an anther lobe remains connected. Right? Remember this figure? Yar bhitor ki thakibo? Microspores bila thakibo na thake. So microspores yato thakibo, sobote thakibo. So gutei khini jodi connected hoi thake, etu kami ki bili kom? Paulinium. Understood? So all the microspores of one lobe will remain connected, and that is what is called as pollinium. Clear? So pollinium, what is pollinium? Question nahe. Pollinium is seen in, the very commonly asked question is about orchids. Right? So orchid or pollinium we need to remember. Next we have something called as a translator apparatus. Translator apparatus are structure like this. Translator apparatus of key association. We have talked about a pollinia, right? Now we are saying that two pollinia of adjacent anthers fuse together to form an apparatus, and that apparatus is called as a translator. Pollinia ki bostu? Jimanbo microspores ase ekelege log lagi ase in each lobe, right? Adjacent anther, we know that in one flower not necessary it will have only one anther. In morphology we have already discussed, no? Dithecous condition, monothecous condition, didelphous, monodelphous, polydelphous, we have talked about all those, right? So in a flower there could be more than one anther. Eta anther of bhitorot ki thakibo? Pollen grains thakibo. So duta if there is an anther here, there is an anther here, yatu pollinium form koribo pare, yatu pollinium form koribo pare, duita jodi log lagi thake, that apparatus would be called as what? Translator. Two pollinium of adjacent anther join together to form an apparatus, and that apparatus is called as translator. What is a translator apparatus? Questions nahe. Okay? Just what we need to remember is, right? Pollinia related question hoda orchid or ahe. Jedi translator related question ahe, teta calotropis or ahibo. Right? Okay, the next point. Clear, no? So we have a term called as polyspory. What is polyspory? 1 mmC forming more than 4 microspores. The phenomenon is called as polyspory. One example is Cuscuta. What is Cuscuta? It's a parasite. What type of a parasite? Stem ne root. Stem parasite. What type of a stem parasite? Total ne partial. Total. Right? Okay. Next. Ebila ki man important no hoy. These are all GK type. We know about a pollen grain, its haploid, its uninucleate, etc., etc. What is important is point number 5. Point number 5 very important. Why is it important? What is so unique about point number 5? What is so exception? What happens to the microspores after they are formed? We know from 1 mmC there are 4 microspores that are formed. What will happen to the microspores? They will germinate. Keita microspore germinate koribo out of 4? Normalot keita kore? Okay, you have heard about the words microspores and megaspores, right? From 1 mmC, either it is microspore mother cell or it is megaspore mother cell, each will divide and ultimately they will form a collection of 4 microspores or 4 megaspores. Commonly, out of the 4 microspores, how many germinate, and out of the 4 megaspores, how many germinate? Gotegita. That is the point that you need to remember. In general, out of all the 4 megaspores, no, it is only 1 that remains functional and 3 degenerate. We have not done it here, but we will be doing it. Diagram something like this. The 3 degenerate korisa, eta okol functional thake. In case of what? Megasporogenesis, right? Megasporogenesis mane ki? Formation of megaspore. Keta form koribo? Sarita. Keta functional hoi deibo? Eta. Right? In case of microsporogenesis, keta microspore form koribo? Sarita. Keta functional hoi deibo? Gute gita. All the 4, in general, all the 4 microspores that are formed will remain functional, but this is an exception. Understand? Normally, Baki Bilak family, all the 4 microspores will remain functional. In Cyperaceae, only 1 will remain functional, almost like megasporogenesis. Clear? Why is this important? Because after this chapter, we will have to do certain types of questions that has been asked. Have you gone through such a question where it has been asked as to, the question goes like this, what is the number of fruits that is formed from iman pollen grains and iman eggs? But what is the number of, if suppose pollen grains iman khini ase, eggs iman khini ase, what is the number of fruits formed? But iman khini fruit form koribo lagile, what is the number of pollen grains or eggs that is required? There are certain, that type of a question that is related from this chapter. From that point of view, we need to remember this. If the question is specially asked about Cyperaceae, a point to monitor rakhibo laibo. Rest of the families will always consider that from 1 mmC 4 microspores will be produced and all the 4 microspores will be functional, so the calculation will go on that line. But if the question is asked specially about Cyperaceae, the point that you have to remember is 1 mmC will produce just 1 microspore. Right? 1 microspore produce korise boli kiyo koise, because 1 is functional, no, baki kita to moriye jai. So 1 mmC produces 4 microspores in all the other families. 1 microspore mother cell produces 1 microspore only in case of Cyperaceae, right? So a point to rakhiba, yar related nami questions korim and then it will become more clearer, right? Next, this is something we know. Yate kiba ase niki? Nai no? Exine and intine. If you are talking about the pollen has a pollen wall and wall cannot ki thakibo, pollen grain not ki thakibo, exine or intine. So one point about exine and intine. Exine is important because that ki thake sporopollenin thake, and sporopollenin related question is who secretes sporopollenin? It is the tapetal cells. Very good. Okay. Okay, Sporoderm, no? So, eta pollen grain or duta wall thake, no? You have the intine. You have the exine, eto to ami janu. Eto jodi pollen grain hoy, pollen grain e bhitorokon ki hobo? Intine ekhon ki hobo? Exine, duikhon collectively called as sporoderm. Derm mane ki outermost layer, right? But skin. So pollen grain to skin buli kole exine and intine together is called as sporoderm. যেনেকৈ আমাৰ ইনাৰ মেমব্ৰেইন অফ নিউক্লিয়াছ আৰু আউটাৰ মেমব্ৰেইন অফ নিউক্লিয়াছ টুগেডাৰ আমি কি কৈছিলোঁ নিউক্লিয়াৰ এনভেলপ ৰাইট চিমিলাৰলি both of them together are called as sporoderm. Okay, who forms the intine? Male gametophyte self. And ultimately intine ki form korbo pollen tube, right? What is important is the word pectocellulose. Pectocellulose to question ahe. ठीक है सर, intine or chiefly composed of remember the word germ pores? Germ pore ki hoy? It is the area where the exine is not continuous, the intine is not continuous. The exine is not continuous. Second point, it is that location through which dash comes out. The pollen tube comes out, right? Pollen grain to short ke likhisu, ha? Pollen grain aro. Okay, ইয়াতে অকমান extra দিছো ইমান বেছি important নহয় ঠিক আছে so we know something called as a germ pore. What is a germ pore? This is that area where the exine is either thin or sporopollenin nathake or the exine is absent. That area is called as a germ pore. Generally we use germ pore is nothing but a pore, right? Depending on the structure of a pore or how does the pore look like, we have two terms. If the pore is round, it is called as a germ pore. If it is a slightly elongated structure, then it is called as germinal furrow. What is germinal furrow? Iman important no hoy. We'll be confined with the word germ pore itself. Germinal furrow you don't have to remember. Germ pore or germinal furrow almost it's the same except what is the difference? The shape. At a round hobo, at a elliptical by elongated hobo. Next, together with pollen grains, we hear certain words called as tricolpat, monocolpat. Is the words related with what? Related with the number of germ pores or number of germinal furrows that is present, right? Tricolpat pollen grain, tetracolpat pollen grain means what? Saita ki thakibo, germ pore thakibo, right? Tricolpat, tinta germ pore. If you are talking about monocolpat, you can either remember it as one germinal furrow or you can remember it as one germ pore. It's the same thing, right? So we have a term called as pollen viability. What we understand by the word viable, the tendency to germinate, right? So jetialake tar germination capacity to thake tak ami ki boli kom viable pollen, it can germinate itself, right? So talking about different plants to plants, their viability period is different, right? So if we are talking about rice and wheat, the pollen grain will remain viable only for 30 minutes after pollination. Pollination hoar pisot kiman deri functional hoye thakibo, only 30 minutes. So it has to get pollinated within 30 minutes or else it will become non-functional, right? Similarly, in case of certain families, they will remain for months, and in pollen banks, you can keep the pollen grains for years together by a process called as cryopreservation. And whenever we talk about cryopreservation, we remember liquid nitrogen, right? Yeah, -197. It's the food. The food. That is a positive aspect, this is a negative aspect. So out of this, you don't have to remember all this. What is important is just etar upor question I had, that is Parthenium. Apart from Parthenium, we have something other pollens also. So pollens are responsible for causing pollen allergy. What is important? Hay fever. Details about hay fever, who causes hay fever, eku nalage. Question 2 ahise, hay fever is caused due to. Hay fever is caused due to pollen allergy. That is the only sentence that you have to remember. Right? A bemar to kihor karne hoy, pollen no karne hoy. Right? Baki details not important. Can I wrap this part? Parthenium is Congress grass ba carrot grass. The most, the major contributor for allergy. Rastai ghate jongholor nisake hoi thake, right? So tar aru eta ki mon rakhibo lage, not indigenous to India, India hua nasile no, it was okay, it came with something, wheat jetia ki korisile, import korisile no, bele countries or pra tar logote ki ahisile, yar seeds bilak ahi goisile, ahar pora it got circulated in India. Now it's become a menace. Right, control koribo nwara obostha imane ase. Kiman asile point number 14. Eta 15 asile niki pollen 14, 18. So we have certain positive aspect of pollen grains also. Various pollen grains are used for the manufacture of certain types of medicines, cosmetics, food supplement, etc. Why is it used as a food supplement? Because it is rich in nutrients. As far as the percentage is concerned, not very important. Carbo is carbohydrate. Online students, can you hear me? Hello? বন্ধ হৈ গ'ল Hello students, Puna paisane? Okay, it got disconnected or what in the middle? Okay. So these are the certain points that are related with the pollen grains, right? We have talked about microsporogenesis, and within the process of microsporogenesis, what we have seen that one MMC give rise to 4 microspores. The microspores remains arranged in a particular type of arrangement, and all the microspores remains connected with each other, and they get separated before the process of pollination. Now we will be talking about the development of the pollen grain. Pollen grain etyalake ami ki poriso, pollen grain form korise, right? Pollen grain to keneke develop korise, keneke germinate korise. This is the thing that we are going to study. So heading koriba development of the male gametophyte. Now what do you understand by the word male gametophyte? First time to hoisile niki? Nai wa. Likha. Okay, ekini hol niki? Etia, before going into discussion, just tell me what is the meaning of male gametophyte? Male gametophyte or development পঢ়িম কিন্তু male gametophyte কি বস্তুটো জানিব লাগিব ন? So what is male gametophyte? It's a pollen grain. It is the haploid stage that produces the male gamete. You are correct, but please give us simpler meaning for that sentence. Iman sentence mukhosto koribo nalage no, simple ke buji poa. Male gametophyte mane ki? Buji paisa ne? Haploid state buli koisa thike ase, tar ploidy to haploid e hoy, thik ase? Male gametes thake hetu correct tumar. Kintu what is the other name for the male gametophyte? হেতু বস্তু বুজি পাব লাগিব, বাকি কিটা সেন্টেন্স তোমাৰ কারেক্ট আছে, ইনফরমেশন ইজ কারেক্ট, কিন্তু মেল গ্যামেটোফাইট মানে কি ইমান কমপ্লেক্স কে নপঢ়িবা হা, কমপ্লেক্স কে পঢ়িলে সব বস্তু পাহৰি যাবা। Male gametophyte is nothing but the pollen grain itself. Gametophyte means what? The gametes bearing plant. Which gamete are we talking about? We are talking about the male gametes. Male gametes kot thakibo inside the pollen grain. So the pollen grain itself is called as what? Male gametophyte. So how does the male gametophyte develop? So pre-pollination, post-pollination to Zanu, is it over? The size of the pollen grain obviously remains the same. In my diagram, eta dangor eta horu hoise, right? That does not mean the shape will keep on changing. Yes, sir. Just a rough diagram, just to understand the process. So, we are talking about the development of the male gametophyte. Male gametophyte is equal to the pollen grain, right? 1 MMC produces 4 pollen grains, and the pollen grains will germinate or they will develop. In the process of development or germination, we can divide the entire series into 2 steps: pre-pollination and post-pollination. Pre-pollination or ki hoy or post-pollination or ki hoy. If you talk about stages during pre-pollination, what is the thing that we see? Starting from the pollen grain, etu ki hoy? Pollen grain or microspore, right? Microspore is 1 cell and 2 cell. Obviously 1 cell because it has 1 nucleus. So this cell will ultimately divide into 2 cells, and what are the 2 cells? The tube cell and the generative cell. If you talk about division from the diagram itself, it is clear that equal division nai kora. One is smaller and the other is bigger. As far as the generative cell is concerned, it's the smaller part and the larger part is the tube cell. So ultimately from this cell, it got divided into 2 cells, and what next? Little later we see the deposition of callose in and around the generative cell, and whenever callose gets deposited in and around the cell, it breaks the connection. At this point there were 2 cells, both the cells were attached with each other, right? Next, if callose gets deposited in and around the generative cell, it will make the generative cell free from the tube cell. So ultimately in this diagram, I think you can see the difference. Yate ki asle, in this diagram also you see the tube cell and the generative cell. In this diagram also you see the tube cell and the generative cell, but what is the difference? Key difference. Yato duta cell ase, tato duta. If you are comparing diagram number B and diagram number D, difference to ki? It looks as if the generative cell is submerged or it is floating inside the tube cells. Yate eta cell directly dutale divide korise. Yate it's almost 1 cell. It looks like 1 cell, within 1 cell there is another cell, right? The generative cell is within the tube cell, right? So up till here, these are the events that occurs during or pre-pollination development. Once it gets pollinated, what happens to the pollen grain? The pollen grain reaches the stigma, right? Pollination mane ki? From the anther to the stigma, pollen grains bilak jabo. If the pollen grain reaches the stigma, kenekwa condition or goise? Remember we have talked about something called as a 2-cell condition and the 3-cell condition in case of Cycas and Pinus. Remember we have talked about that, pollinated in a 2-cell stage, pollinated by a 3-cell stage. If you are talking about pollination of pollen grains, normally in majority of the cases, they get pollinated in 2-cell condition. Majority of the angiosperms, around 70% or 80% of the angiosperms or ki hoy, 2-cell condition or pollinate hoy. Certain types of angiosperms we see 3-cell condition, and if you are talking about 3-cell condition pollination, tetiyale ki hobo, generative cell will get divided and form 2 male gametes, right? Eta cellote ako 2 cell form koribo. So either it can be pollinated by 2-cell condition or it can be pollinated by 3-cell. Here what we are talking about is 2-cell condition. Pre. After that, once the pollen grain falls into the stigma, it starts absorbing the water that is present in the stigma and it swells up. If it swells up, what happens? The intine comes out of the germ pore and ultimately they form the pollen tube. What is important about the pollen tube? The question has been asked about the position of the tube nucleus. A2 cell or pra, A2 diagram hoise, intine or pra ki form korise, pollen tube to form korise. So obviously, a generative cell to keyhole, it got divided and it formed 2 male gametes. What will happen to this one, right? So the question has been asked about the tube nucleus. The tube nucleus later gets degenerated, the first point. Second, what is the position of the tube nucleus? The tube nucleus is always found at the tip of the growing pollen tube. Hey, tip word to monitor rakhibo lagibo. Thik ase, tube nucleus to kot paba, ebilakot paba, net tip word paba. It's always on the tip. And we know the pollen tube at the very starting point itself, it was not as long as it's shown in the diagram, no? Horu horuke asle, horu horuke digholo hoye goye ase. So tipot ki thakibo degenerated. Next, talking about the pollen tube also, we have certain points to write about the pollen tube. I think we can likhiu dibo paru. So pollen tube what ki thake, we have seen certain things called as callose deposition or callose plugs, right? Now you have to know what is the function of the callose plugs. The function of the callose plugs is it divides the old cytoplasm from the recently formed cytoplasm. old cytoplasm, recently formed cytoplasm. As the pollen tube is enlarging or it's becoming long, tat ki hobo cytoplasm increase hoy goi thakibo na thake. So agote hua aru notunke hua cytoplasm difference ase. So notunke hua cytoplasm khini khodai it will be this part and it will remain active because the male gametes are present here. Right? Ekhini loke jimanbor cytoplasm asile, all these are considered as inactive cytoplasm. So who separates the active and inactive cytoplasm in the pollen tube? It is the callus plugs. Right? Next, if you are talking about the male gametes, duta male gamete to thakibo, we understand, and the male gametes are surrounded by a thick layer of cytoplasm. Right? So in the pollen tube, we have certain zones, jeneke vacuole zone, vacuolization, vacuole ase no? So vacuole ase karone to dangor hoy. Eta cell grow keneke kore. I mean, credit goes to the vacuole of the cell also, because the vacuole will enlarge and ultimately the cell will also enlarge. This is something that we will study in plant growth and hormones boli eta chapter ase no, tat ami cell growth keneke hoy, we will talk about that, right? So this is all about the diagram. Ekhini bostu we are not going to write in sentence because it's well understood what are the events that's taking place. Ki monot rakhiba? Equal distribution no hoy, unequal division of cells, smaller and a larger cell. Who forms the male gametes? What happens to the tube nucleus? What is the position of the tube nucleus? Right. I think the time is almost over. So what we'll do is, after this jikini points ase, we'll do it in the next class. Okay. Thank you. Oh, nachano. Yeah, right.